So hi there. Today we are going to talk about like a little bit on PRSice, and because it's a simple, relatively simple software, we'll also talk about some of the basics of polygenic score analyses. So my name is Sam Choi. I'm from O'Reilly's lab from Icahn School of Medicine at Mount Sinai. Now, first thing first, we have to go through like the fundamental of polygenic score. So polygenic score can be calculated as the sum of betas times x divided by n. The betas are the effect sizes and the x are the individual genotypes. So essentially, it's like the weighted score like of your genetic dosage. You can notice that like there's an n here, which is the number of alleles. This helps us to account for the number of missingness, and it's slightly different from what you usually see in polygenic score papers. So like to illustrate, assume that we have 3 individuals that each have a different alleles. Some of them might be missing. And then each of those, we have something called the effective allele. So for example, for SNP A, the effective allele is A. So what we are saying is that for allele A, it has an effect size of 0.1 relative to the reference, which is like the non-effective allele, which is the allele G. For SNP B, the effective allele is G, and then it has an effect size of -2. So effect size doesn't have to be all positive. It can be positive and negative. And then we have SNP C, which has an effective allele T with a value of 4, which is like huge. But anyway, let's go ahead and see how we calculate the polygenic score. So for each individuals, we can basically we can try to multiply the alleles with the corresponding effect size. So for individual A, it has 2 effective alleles. So we will times the effect size by 2. So we get a 0.2. For the second individuals, it has 1 effective allele. So we'll say that it has 1, we'll multiply the betas by 1, so 0.1. And the third individuals has no effective allele. So all of this allele is like non-effective, so it has a score of 0. Now we go ahead to, excuse me, to the second SNPs. It's almost the same, but when we have missing data, we'll usually just apply a 0 to it, or sometimes we'll do a mean imputations, which use the population's allele frequency of the effective allele, and to mount in place of like 0. So if this is our populations, for example, we'll say that maybe we have like 0.25 allele frequency for G across, because we only have 2 samples and there's only 1 copy of alleles. But here we'll just say 0, and then so on and so forth. What we'll then do is we try to sum up every single one of this score into one single total score. So for individual A, that would be 4 times 0.2. So 4.2 divided by the number of alleles. So 1, 2, 3, 4, 5, 6 alleles. And same for the second one. But for the last sample, because we have 2 missing alleles, when we divide the numbers, we'll say we'll divide it by 4 instead of 6. Now, if we do mean imputations, we'll still do it by 6. So that's like some of the fine control of the allele frequency, like how you do missingness imputations. And some software like PRSice do have like functions, like mPlane do have functions for you to have a more fine control of how to do like this kind of imputations. And some other software will just always imply a 0 score. So after that, we can just calculate the score. So we can say that the first person have a score of 0.7, the second has a score of -0.317, and the last person has a score of 2. So this is the like general concept of how a polygenic score is calculated. Now, there are actually alternative formulas for PRS calculations. One of the more common one is sum score instead of what we are using, the average score. The main difference is we we remove the denominator from the equations, and this is usually what was shown in the literature. The second one, oops, why is it not? So the problem of this sum score is that it can be biased by the level of missingness. So if, for example, we have a sample, like there's an individuals with a large amount of individual genotype missingness, then by definitions, that sample's score will be relatively smaller than other individuals. And when you try to rank samples or try to distribute them across the populations, this will introduce a bias that is like correlated with the level of genotype missingness, and that might not be ideal. That's why, like for PRSice and Planck, the default settings is the average score, not the sum score. Now, another very popular formula is the standardized score. What it does is take the average score and look at the whole populations, and we try to standardize it so that it has a mean 0 and standard deviation of 1. The problem of this standardized score is that for binary traits, for case control samples, when you standardize your score, it can be affected by ascertainment. So imagine in a data set where you have like 50% cases and 50% control for a trait with population preference of 1%, your data is enriched with cases. So when you calculate the mean and standard deviations, the cases contribute more than what you will otherwise observe in the population. And this introduces a bias. It's not as much of a problem for quantitative traits, so that's more specific for case control. In PRSice, we have an options where you can standardize your score using only the controls. We assume the controls are more representative, more random. That is something that you can do, and something that you should consider when you do the analysis. Now, as I just mentioned before, there's also different way of handling missingness. Now, there's the mean implementations that I just mentioned, which you can use the allele frequency of your reference genome or your target sample data to estimate the minor allele frequency. The other one is zero-centered. So you try to make sure that the missing sample all have a value of 0 by minusing all the scores by the allele frequency. So it's similar. Now, the main challenges in polygenic score analysis is actually that we are mainly concerning two factors. The first one is the Wiener's curse. When you select allele SNPs or variants for PRS constructions, they were selected because usually because they're significant or because they have a high effect sizes. And that, unfortunately, when we do selections, those that were selected usually have an inflated value. That's why they got selected. And this is something we have to account for when we do the analysis. Another problem is linkage disequilibrium. When we do GWAS, linkage disequilibrium help us a lot because now we can reduce the number of tag SNPs that we need and we can still have a good coverage of the genome. But when we calculate a polygenic score, if we have multiple alleles in the same regions in high LD, then the effect size of that particular allele will got enriched and calculated many times, and that will cause a bias. Now, to deal with Wiener's curse, in polygenic score, we usually do something called shrinkage. What we essentially does is we push down the effect sizes by a certain factors using different methods. And we are saying that by shrinking or by pushing down these effect sizes, we get a better representations of the true effects, and hopefully we'll get a better estimate. Another one is linkage disequilibrium. GWAS relies a lot on tagging, so we doesn't necessarily have genotype the causal variance, and it makes it quite difficult for us to say this variant is the true causal variance, and we can just guess. So and variance, this does lead to the situations where when we calculate polygenic score, variance in LD to each other can lead to double counting. So imagine the most extreme scenario when two alleles are in perfect LD, two variants are in perfect LD. And you don't when you calculate the polygenic score, you're essentially calculating the same score, like same alleles twice, putting in the same model. Now, if you imagine there's a region of the genome that are in high LD, like chromosome 6, like MHC regions along LE, then you will get your score would be dominated by this kind of regions. And you don't want that because that causes bias. So we introduced a method called PRSice, or more like a software called PRSice. It's a simple approach to polygenic score analysis. It mainly uses two methods, the clumping method for accounting for LD and the p-value thresholding method as a form of shrinkage. It's computationally very efficient, and relative to other software, it's slightly easier to understand and comprehend, although it does have a lower performance in terms of R-square and p-value of associations. But we do argue that this is a very good starting point for you to get hold of your data, get a general idea of how your polygenic score will look like, and then improve upon maybe using more complex, more advanced software like lassosum, LD per SPACE-R, and PRSice, all of which you might have exposed to in the PGC day. This was first developed by Jack Houston in King's College London and is now maintained by me and Dr. Paul O'Reilly. So with linkage disequilibrium, PRSice performs something called clumping. So imagine we have a bunch of variants. I use this crappy DNA symbols to represent variants, and I rank them by their significance. So the taller they are, the more significant they are. They are in LD, so they're linked together. Now, if I don't do anything and just calculate the score, then like this alleles can get double counted or give a high proportions of effects to the polygenic score. What PRSice does is that we look at the variants. We select the most significant SNPs or variants, and then we remove any variants that are in LD with it and have a lower, less significant p-value. So here only the pink variants will remain in after clumping, and those will be used for polygenic score calculations. Next up, we do p-value thresholding. So imagine we have a GWAS, like this is, I think this is the PGC 108 schizophrenia GWAS. What we do is that we will perform some kind of p-value thresholding. It's a way of hard thresholding. What I meant is, let's say we only select SNPs that has p-value less than 1e-a. It means that any variance that has a p-value higher than 1e-a, we'll get the effect size shrank to 0. So the effect size becomes 0. PRSice will iteratively change the p-value threshold. And then as you can see, we retain different portions of SNPs in the polygenic score. And we shrank the rest of the score variance to have an effect size of 0. The way PRSice select the p-value thresholding is by means of, at each threshold, we'll get a PRS, and we'll regress it against the phenotype of interest. We'll then say that the regression model that gives us the best performance, highest R-square, is the best threshold. Now, a lot of people complain that this might be overfit. And it does, like, because of this model optimizations, it does cause some kind of overfitting. But this overfitting is inherent to polygenic score model, not just to PRSice. So what we developed is that in PRSice, we try to do something called permutation, like to calculate empirical p-value. So what it does is we first calculate we use the best PRS. We look at like what p-value it does provide us. We then shuffle the phenotype to obtain a new p-value. And we repeat it like 10,000 times. And this gives us null distributions of possible p-values. And then we can then calculate the empirical p-value as the number of time we observe a better, more significant p-value than the observed p-value. This helps us to alleviate a little bit of the overfitting problem of testing many parameters. So that's the gist of it. So before you, we'll go through how, some general principle of running PRSice and some of the things that you need to think of when you do it. So before you start PRSice analysis, you'll need the GWAS summary statistics, and you must have the SNP ID column, the effect size, effective allele, and p-value column in the summary statistics. Those are the bare minimum. The SNP ID is for matching the genotype data with the summary statistic data. The effect size is giving us the effect size estimate. The effective allele tells us which allele it is so that we can do strand flipping. And p-value is for clumping. Now we also have require genotype data for the target samples. And these samples must not be included in the GWAS, otherwise you'll get into trouble. This is fundamental to all PRS method. You must not have any of your target sample present in the GWAS, or you will get highly inflated abnormal results. Now PRSice does accept file in plain or BGen format, so you can just directly use them, which is fine. You can also give PRSice the phenotype data and the appropriate covariates if it was not already included in the FAM file or the sample file. You can also give PRSice an LD reference, 1000 genome, if your target sample size are small, less than 500 samples, although that's usually don't have enough power for PRS, and that might be something useful. Now, PRSice is mainly divided into two parts: the PRSice executable, which is used for the core analyses, and the PRSice.R, which is an R script that is just used for plotting. Before you start, though, you should perform some basic quality controls on your data. For example, if you have any duplicated SNPs in your GWAS, you should make a decision of whether you want to maintain one of those copies or do you want to remove both of those copies. You should also do common GWAS filtering on your target data. For example, remove related samples, remove samples with sex mismatch, remove SNPs or samples with excessive missingness, etc., to just ensure high quality data. And if you want more, you can read our PRS tutorial paper on Nature Protocol, and there are more underlying outline of those. So running PRSice is hopefully intuitive. We do try to make sure that our documentation is clear and our parameters as makes as much sense as possible. So running PRSice is just type PRSice, and then you have to give us the target sample using the B file parameters. You just give us the prefix, and then you can give us the base, which is the summary statistic file, the GWAS. If your GWAS has a standardized format, so for example, the SNP ID is called SNP, the effect size is called betas, p-value is called p, the effective allele column is called A1, then PRSice will automatically detect those. Otherwise, you can always use the appropriate parameters, minus A1 for the effect size, effective allele, minus steps for effect sizes, etc., to indicate, to tell us which columns correspond to which informations. You can also give us the phenotype file using minus pheno, and then use minus pheno column, minus column to indicate which column you want if you have multiple columns. You would then need to tell us whether it is a continuous or binary target using binary target. If it is continuous, PRSice will use a linear regressions to identify the best threshold. And if it is a binary phenotype, then it will do a logistic regression. You can then also give a covariate file and use minus covar, cov minus col to indicate the columns that you want to include in the covariance. And then one of the most asked about features is print SNPs. So people ask us if there's any way to show the SNPs included in the PRS and SNP options is your friend. And finally, to get the empirical p-value, you use minus per 10,000. We will recommend at least 10,000 because that's when you get a relatively stable p-value at 0.05. You can go more, but it's very computationally intensive. If you want to like get the plots, all you need to do is just to add use the R script instead. Sorry, misspell. Should be R script, not R C. Yeah. PRSice.R and then minus PRSice, and then tell us where the executable is, and all the other comments are identical. Now, one of the most important output from PRSice is the doc summary file, which is show us the best model fit. It will has information of the phenotype, the set, like this is something related to preset. Usually it's just base in PRSice, which is genome-wide score. Threshold tells you which p-value threshold we use. PRS squared is the variance explained by only the PRS. Full R squared is the variance explained by the full model, including the covariate and the PRS. And then now R squared is the variance explained only by the covariates. If you have a binary traits, you can give us the preference, and then we'll do an adjustment of the PRS to account for ascertainment bias. If not, then it will just be minus. A coefficient is the regression coefficient. It can be like huge if you use the average score, because the 1 unit change of polygenic score when you take the average and given the small effect size is a big change in phenotype usually. So if you will have a large value for coefficients. If you want it can be smaller, to be smaller than your standardized score will generally give you a smaller coefficients, but it shouldn't change your p-value or the R square much. P-value, P is the p-value model fit, standard error is the standard error of the model, number of SNP is the number of SNP included in the model. And if you do the permutations, then you will also get an empirical P column. You can also look at the individual threshold model fit by looking at the doc PRSice file, but we generally found that not very informative, so we ignore that quite a lot. And then the other important output is the PRSice.best file, which contain the score at the best p-value threshold. It has 4 columns for PRSice. FID and ID are the individual ID provided and usually included in the plain file. In regressions, suggest whether these samples were included in the regression model. So if your sample has a missing covariate or missing phenotype, we will still calculate the polygenic score, but then in this best score folder, we will tell you that it is not in the regression model by having a no in this column. And then finally, the PRS, which is like the polygenic score column. If you use the R script, you will also get some useful plots just for your eyes. So this is like the bar plot, which is quite famous. So it shows you the R square at each different p-value threshold and the corresponding p-value at top, and also the colors. So you can see immediately that the best p-value threshold is 0.4463 here, with an R square roughly 0.05 and p-value of 4.7 times 10 to the minus 18. Alternatively, we also have a high resolution score if you use the high resolution options of PRSice. It shows you the p-value like model fit of each individual p-value threshold. The green line represents the same threshold that were included in the bar plot. And this shows us like where the best p-value threshold is and the corresponding p-values. So common problems we encounter with polygenic score like PRSice, we do try to capture as much of them as possible. So we will warn you about duplicate samples, duplicate variants, missing like excessive missing covariate, wrong definitions of phenotypes, etc. We try to do that, but we cannot detect sample for that. So this is something that you have to do it yourself. But general rule of thumb, if your results are too good to be true, so for example, everything is like very significant, or like the R square is extremely high, or something like that, basically a flat plot, then you know that you have sample overlap, most likely. So this is almost the end of it. If you want to get more information of PRSice, you can go to PRSice.info, which is our official website. Or if you want to know about more general about polygenic score or how to do polygenic score or what are the concepts, then you can go to our polygenic score tutorial in choicing1.github.io/PRS tutorial. So that's it. Thank you very much. And this is the slide. Yep. That's